I was telling you that we just discussed this glycogen phos. We have just discussed this glycolysis step. Okay, we just discussed this glycolysis step, its enzyme and its regulators. Okay, now we because we have just seen the example of glycogen phosphorylase, I will teach you this along with it. Okay, so that we may not I have to then change pages of your Lippincott book, so that is why I am like shuffling it, okay, from your first page. So now while studying glycogen, so you have to the only reason I'm showing you the examples written in your book only because there are other phosphorylase enzymes too, but I'm not using the other enzymes so that I may focus on the thing which is written in your first stage, because your exam questions are like exactly your book words. They don't even change the words of your book. They just extract those things from your book and just make an MCQ and put it in your paper. So try to focus on every word written in your first aid biochem book, and you will not miss a single MCQ of your biochem in your paper. That is for sure. Okay, revise your complete this biochemistry portion from your book, and then revise this first aid biochem like 3 times, 4 times, 5 times, and there will be a time when you will be like revising this whole in like 3 hours or 4 hours from the first page to the last page, everything. Like you will be so quick in it because you already know and the concepts and everything, and you just have to focus on the points that are important. So you have to learn these enzymes, all enzymes and processes. Okay, so learn the glycogenolysis, the breakdown of glycogen or the degradation of glycogen. Rate limiting enzyme is glycogen phosphorylase. Okay, now you understand what a phosphorylase do. It adds a phosphate group into the substrate. Okay, now what are the regulators? Again, very interesting. Why do you need to break glycogen? Because your body is deprived of glucose. When your body do not have glucose, only then you will break a polymer. No, like there are some guests in your home, okay, and you have this whole bucket of ice. Okay, it's not in cubes. It's like you put in water in a tub and you just put that in freezer, and then you have this huge piece or chunk of ice. But now there are guests in your home and you have to um, you know put ice in their glasses, so you need to break them in small pieces. You need to break this ice in small pieces and give 1 cube or 2 cube in one glass. That is what we do. Okay, like when we have this huge big gatherings in our home, we don't put ice cube trays because they have very little space for ice. So we give this whole big bunch of tub and we put it in a freezer, and then we break it and we have this small chunks of ice so that we can put in glasses or so on, or in jugs or something. So the same thing goes into your body. When you have a storage of glycogen, this is a huge, just take it as a huge chunk of ice. When you have this glycogen and your body is deprived of glucose, and your liver wants glucose, your brain wants glucose, your kidney wants, your muscles wants, your every organ that is performing some activity wants little glucose. What you will do? You will break it and you will give one chunk. Now when you break it, this is no more glycogen. This one chunk will be called glucose. You will give this one chunk to some organ and one chunk to some other organ. So the point of discussion is, when you have sufficient amount of ATP, again the same mechanism. When you have a sufficient amount of ATP, lots of ATP already, you don't need to break down this chunk. You don't need, after glycogenolysis, you have glycolysis, and after glycolysis, you have TCA, and after TCA, you have electron transport chain. This is how it goes. You have lots of ATP. You don't need this, you don't need this, you don't need this, you don't need this. Getting my point? You have very little amount of ATP. Or you can say you have AMP or you have ADP. You have low energy molecules. Okay? You have these low energy molecules. When you have these low energy molecules, you need to break down this chunk of glycogen. You need to generate glycolysis. You need to generate TCA cycle. You need to generate electron transport. This is how it goes. So now the same thing applies. You have more ATP. More ATP will inhibit. Negative sign shows inhibition. More ATP will inhibit this glycolysis. More ATP will inhibit this glycolysis, this glycogenolysis, this TCA cycle. Okay, more ATP will inhibit this all. TCA, glycolysis and glycogenolysis. Please remember. And more AMP. Okay, more AMP means less energy molecule will activate glycolysis, activate TCA cycle, activate glycogenolysis. So these two things are same for these three of them. Okay, so like you can see it also over here. ATP, more ATP, more ATP, more ATP. More ATP are inhibiting TCA, glycolysis and glycogenolysis. Okay, and more AMP. Okay, then again more ADP and more AMP. They are activating glycolysis, TCA and glycogenolysis. So this is same because all these are generating energy. More energy, less cycle. Less energy, more cycle. Simple concept. Okay. Now I will make you understand what these other things are doing. If you will understand the concept of glycogenolysis with this, the opposite will be of genesis. Because here you are breaking the chunk of glycogen and here you are forming the chunk of glycogen. Like here you are joining these cubes to make a big chunk and here you are breaking this big chunk into small cubes. So whatever is going on at this level will be opposite at this level. Okay. So try to understand the concept of one and then turn it over for the other one. Okay. So now you see insulin and glucagon. What is the function of insulin? What insulin do? It decreases the level of glucose in your body. Simple. Okay. Insulin is opposite to glucose. Where in diabetic patients who have this problem in their beta cells of pancreas, okay, they do not work. Diabetes type 1 or type 2 patients, when the beta cells of pancreas are not producing insulin, no insulin means their blood has more glucose. Their glucose levels are not controlled. So you give them synthetic insulin in the form of like medicines. Okay, they take these insulin injections. So that insulin decreases the glucose. Okay, so the same concept applies over here. You are running this cycle to have more glucose. This cycle gives you more glucose. And if you have sufficient amount of insulin in your body, do you need more glucose? No, you don't need it more. Okay, because they are opposite. But when we talk about glycogen, when our body has less glucose, glycogen is released. Now your body has sufficient amount of glycogen. Will you generating this cycle? Yes, because glycogen is produced when glucose levels are low. Let's say for example, let me make it even more easier for you. This is fasting. This is a person who is fasting and this is a person who has just taken its meal. Okay, this is a person who has eaten food and his glucose levels are high, and this person who is fasting, his glucose levels are low. Let me change the color. Okay, this person's glucose levels are low and this person's glucose levels are high because he is at well fed state and he is at fasting state. Now in this individual who has eaten food and glucose levels are more, in his body insulin will be secreted. The pancreas will secrete insulin to make this glucose low. Okay, so when the body has when the body is secreting insulin to make this glucose level low, will you be running glycogenolysis? No, you will not be running glycogenolysis. Why? Because glycogenolysis give you more glucose and your body is secreting insulin to decrease the level of glucose. So insulin will inhibit this cycle too. Understand? Insulin will inhibit this cycle so that level of glucose in your blood comes down. That is the function of insulin. But on the contrary, this person who is fasting has low level of glucose in blood and his pancreas are secreting glucagon hormone. The pancreas are secreting more glucagon so that glucose levels become more. So that means your glycogenolysis will run more because glycogenolysis is also making more glucose and glucagon is also making more glucose. So glucagon will trigger this cycle plus and insulin will inhibit this cycle minus. Getting my point? So see this picture. Insulin minus glucagon plus. Glucagon is making more glucose. Glycogenolysis is making more glucose. So glucagon will trigger this cycle. But insulin is decreasing the glucose levels in your blood, and when your insulin is more in quantity, it will inhibit this cycle, glycogenolysis. Okay, so these are the concepts. Okay, but now when you compare glycogenolysis with glycogenesis, you see the opposite is going on. Opposite is going on. Here insulin is inhibiting, here insulin is promoting. Here glucagon is promoting, here glucagon is inhibiting. Because glycogenolysis is giving you more glucose, but glycogenesis is converting glucose into stored form, that is glycogen. So glucose levels will go down and glycogen levels will go up. So when it is opposite, the regulators will also work opposite. So remember one and apply that concept to the other one. Okay. Now we are left with glucose 6-phosphate. You see, glucose 6-phosphate is inhibiting glycogenolysis and glucose 6-phosphate is promoting glycogenesis. The same things are going here also, opposite work. So we will see glycogenolysis. Okay. Glucose 6-phosphate is a product of glycolysis. You have glucose. You have glucose 1-phosphate. You have glucose 1-phosphate. You have glucose 6-phosphate. This is your glycolysis. Okay. Glucose 6-phosphate. Okay, and then fructose 6-phosphate, and then glucose 1-phosphate, and then glycogen. Okay, now it is correct. So what is going on in this picture which I have drawn? This is your glycogenolysis and so on. Okay, and this is your glycolysis. Okay, so they are interconnecting with each other at this step. They are interconnecting. Okay, so this glucose 6-phosphate inhibits this cycle. You understand why you have more glucose 6-phosphate? You don't need this conversion. You don't need this conversion. You don't need this conversion. You have less glucose 6-phosphate. You need this conversion. You want to make more glucose 6-phosphate. So when you are having, when the product is more, the cycle stops. This is called feedback inhibition. The product is more. The product is more. Okay, the product is more. The cycle will stop. Feedback inhibition. The product is less. The cycle will occur more. Okay, feedback activation. You will be studying this feedback inhibition and activation in endocrinology a lot. So understand this concept here. Okay, so you see, glucose 6-phosphate more, feedback inhibition. It will inhibit this cycle. Glucose 6-phosphate is more. It will promote the formation of glycogen. The glycogenesis will perform more. So I don't know if you're understanding me or not, Manzoor and Marcella. Please tell me in the chat section that are you understanding what I'm trying to explain you? Like I'm repeating multiple times so that you may understand these concepts. If yes, then we will proceed. Okay, very good. What about you, Manzoor? Now we are left with epinephrine concept only. Okay. Okay, very good. Very good. So now what is epinephrine doing? What is epinephrine? Epinephrine is a neurotransmitter of a sympathetic nervous system. Okay. When your body is in flight or fight, that is your sympathetic, right? Your body is in like hurry. So when your body is in flight or fight state, what happens is your you need more and more glucose. That is, I can say you need more and more energy. In fight or flight state, you need more energy, you need more glucose. So you will be promoting this glycogenolysis because it is giving you more glucose. Okay, and opposite thing will happen with glycogenesis. You already have more energy. Now in this, you already have more epinephrine, you have more energy. So epinephrine will inhibit this cycle. It will say, please, I don't want glucose. Store the glucose for future, for the bad times, please make and store the glucose. So epinephrine will inhibit glycogenesis, the formation of glycogen. Okay, but epinephrine will promote glycogenolysis because epinephrine is secreted in a fight or flight in sympathetic nervous system, in sympathetic activities, and we need more energy in sympathetic activities. So we will be breaking this chunk into smaller pieces of glucose. You get my point? So this is how we work. And one more concept is here for cortisol. We are left with this hormone here. So cortisol is a hormone which does this opposite work. Okay, from where cortisol is secreted? Anybody? From which endocrine gland cortisol is secreted? Any one of you know? We have this adrenal medulla. We have this adrenal gland. This is adrenal cortex and this is adrenal medulla. The outer part is cortex and the inner part is adrenal medulla. Okay. In this adrenal medulla, I will divide this adrenal into three parts. We have three zones. Okay. There is this zone which is called zona fasciculata. Okay. We have zona glomerulosa. I I there is a mnemonic in your first aid book, very good mnemonic. Okay, let's say this is your adrenal gland. I am dividing this into three parts. Now this yellow one is one part, this green one is the another, okay, and then this dark blue one is another. So this all is your adrenal medulla. So this part is G, this is F, and this is R. Very easy to remember. Okay, very easy to remember. How? GFR. GFR. We study GFR in renal, right? GFR rate. So you can learn it by this GFR. These are zones. So this G is glomerulosa. This F is fasciculata, and this R is reticulosa. Okay, zona glomerulosa, zona fasciculata, zona reticulosa. So now we have this GFR. This F region, this middle portion secretes cortisol. Please don't forget, these are your free MCQs. Okay, this is your endocrine lecture actually. Okay, so this red portion is your adrenal cortex. Okay, I made it like this, right? So I have removed this portion. I have removed this portion from center, and now this is your adrenal cortex. Adrenal cortex have these three zones, not medulla. Please don't confuse. Okay, I have removed this medulla part from the center, and this is adrenal cortex. So adrenal cortex have these three zones, and zona fasciculata secretes cortisol. Okay, please remember. Now, what cortisol do? The important thing. You can like understand it from here. Cortisol is triggering the formation of glycogen. From glucose, you are making glycogen. Cortisol is triggering this process. It means that cortisol is secreted when your body do not need glucose, when your body is like in well-fed state. Okay, when you are not starving, when you are not hungry, at that time cortisol is secreted. Cortisol has other functions also, but one of the functions is the formation of glycogen. Please remember, do not forget. Okay, this is one function. Cortisol has many other functions also. Okay, it is a glucocorticoid. It gives energy to your liver, to your adipose tissues, to your muscles. Okay, and WBCs also, etc., etc. It has many functions, but please don't mix it up right now. Just remember that cortisol promotes the formation of glycogen. It is secreted when you are not starving, when you are in a well-fed state. Okay, please remember. So now we are done with these things. Okay, we are done with glycolysis, we are done with glycogenesis, we are done with glycogenolysis. You understood the regulators, you understood the rate limiting enzymes. Okay, please remember the rate limiting enzyme for glycogenesis is synthase. There is another concept which I want to tell you. Very nice thing to remember. Okay, one second. Let me. Okay, just see what I'm trying to explain now. I'm telling you a way to learn. Okay, now this is a very nice way to learn. Synthase and synthase. You have in this whole list, you only have two enzymes which end with synthase, and both of their cycles have genesis. So it is another easier way to learn that glycogenesis has glycogen synthase and ketogenesis has HMG CoA synthase. Because when you will be studying these two, ketogenesis and cholesterol synthesis, the names are same. HMG-CoA synthase, HMG-CoA reductase. So then you will be confused कि synthase किसमें था और reductase किसमें था. So easy to remember. The Genesis one have synthase. Okay, the Genesis one has synthase. So please remember. Genesis has synthase. Genesis has synthase. Okay. Same, you see the names are same, glycogen synthase and glycogen phosphorylase. But the Genesis one has synthase, the other one left has phosphorylase. So even now, like I am explaining you so conceptually right now. So even if you forget the concepts or at that time students panic, exam में panic हो जाता है बंदा. So at that time you have to use your memory. So you can like use this memory that this enzyme has the same name and these two enzymes are also having a same name. Exactly same. Only the last words are different. So here you have glycogen synthase, here you have glycogen phosphorylase. Now you will be confused कि glycogenesis में था या glycogenolysis में था. So please remember Genesis has synthase. Okay. Same thing with here. HMG-CoA synthase, HMG-CoA reductase. तो अब किस वाले में क्या था? So Genesis वाले में synthase था. Okay. So remember like this. Very easy to remember. And remember the glycolysis with PFK1. It is a very different hormone and please remember it. Similarly, gluconeogenesis. Remember fructose 1,6-bisphosphatase. Okay. Now we will see gluconeogenesis. Okay, we will see what phosphatases do. Okay, let's go back up. So now you see you're done with synthase, you're done with kinase, you're done with phosphorylase. Okay, now phosphatase. So phosphatase is also dealing with a phosphate group, but it is removing, it is not adding. Okay, so what in which step you will see this? What will you see? What is fructose 1,6-bisphosphatase doing? Let's see this beautiful step. Okay. See this step. This is a picture from your first grade. Okay. Just see this little that I have zoomed. Okay, on your mobile screens you can see this zoomed version. So now this step in which you are converting fructose 6-phosphate into fructose 1,6-bisphosphate. In this step you are using this number 8 is fructose 1,6-bisphosphatase. Okay, if I share you the key with you, this is the key. Okay, if I share this key and you see this little step, okay, you see these two enzymes. This is fructose PFK1 is the enzyme of your glycolysis. So I told you glycolysis is like going down and gluconeogenesis is going up. Okay, so glycolysis is using this number 7 in the same step, like you are converting. I'm clearing this and I'm making it bigger because I want to write. Okay, now in the same step, like this is the same cycle, you're converting glucose to pyruvate and you're converting pyruvate to glucose. So while dealing with glycolysis, you are converting this fructose 6-phosphate into fructose 1,6-bisphosphate. Okay, and the enzyme is PFK1 when you are talking about glycolysis. Okay, but when you are talking about when you are talking about gluconeogenesis, you are going like this. You are going upwards. You are converting fructose 1,6-bisphosphate into fructose 6-phosphate. And your enzyme is fructose 1,6-bisphosphatase. It is not PFK1. So, you see, arrows makes enzyme different. When the arrow is down in the same step, the enzyme is PFK1. When the arrow is upwards in the same step, the enzyme is fructose 1,6-bisphosphatase. Why it is like this? Why do you think it is like this? Okay, so let me. So now you see, this is your fructose 6-phosphate. One arrow is going down, and you made fructose 1,6-bisphosphate. Okay, and the enzyme you used was PFK1. What is this down arrow? You are doing glycolysis. Okay, now I'll change the color. Here you are doing gluconeogenesis. Okay, and the enzyme is fructose 1,6-bisphosphate, and the arrow will go like this. Same steps, same things. Enzymes change. Kinase was responsible for adding a phosphate group from a high energy molecule. ATP was giving phosphate. The phosphate was present at carbon number 6 position, and then in this product the phosphate is present at carbon number 1 and carbon number 6 position. So 6 तो वैसे ही है. The 6 is the same. The phosphate you are adding via ATP. ATP is giving the phosphate. This phosphate goes to one number. So that is why you say kinase is working here. But over here, what is going on? At this level, you see this phosphatase. Phosphatase is removing phosphate group, and your arrow is going upwards. So you are removing this phosphate present at carbon number 1. You are removing this phosphate and you are only left with one phosphate that is at 6th position. Are you understanding my point? You removed one phosphate from this, and now you are left with one phosphate at 6th number. Earlier you have phosphate at 1 number and 6 number. So this enzyme phosphatase removes phosphate group. And kinase add a phosphate from a high energy molecule. You were adding it on number 1, and this phosphate was coming from ATP, so the enzyme was kinase. But in gluconeogenesis, you are removing one phosphate, okay, from this 1 number, and now it is only at 6 number, and the enzyme was phosphatase. So this is the thing that is written in your book. So in your textbook, phosphatase, in your textbook, phosphatase is removing phosphate group from a substrate, and you have just seen the example. Okay, please remember that gluconeogenesis rate limiting enzyme is fructose 1,6-bisphosphatase. Okay, if this enzyme will not be present, the gluconeogenesis will not occur. You will not be able to form or produce or synthesize glucose from proteins or lipids. Okay, please remember. So now the regulators are AMP is less. Okay, so that means when you have now, when you were running glycolysis or TCA or other cycles, you were dealing with, like, you were making more energy, making more ATP. Okay, but when you are going opposite and you are dealing with gluconeogenesis, okay, you don't need ATP, you don't need energy. Okay, so what will happen is, in this step, AMP, low energy molecule, will inhibit this pathway. Low energy molecule will inhibit this pathway because we are going towards low energy. Okay, we are using more energy, high energy molecules, we are using ATP, we are using ADP, and we are creating AMP. But when your body has enough AMP, you don't need gluconeogenesis. Okay, you do not need it. So please understand the regulators terminology. Okay, and then I have already told you fructose 2,6 bisphosphate is a product of your gluconeogenesis, and when you have this already, you don't need to run this cycle. So these are very important. Please remember the concepts of glycolysis, gluconeogenesis. Okay, we have studied 4 cycles, we have studied 3 enzymes: kinase, phosphorylase, phosphatase. Okay, from here and from below, we have studied glycolysis, gluconeogenesis, glycogenesis, and glycogenolysis. Okay, now let's see TCA cycle. In TCA cycle, you see this enzyme dehydrogenase. Okay, so let's work on it. First of all, you should know what dehydrogenase is doing. Dehydrogenase. What is the function of dehydrogenase? Dehydrogenase catalyzes or speed up oxidation reduction reaction. You have always heard redox reaction. So dehydrogenases work on redox reaction. What is a redox reaction? Either you are losing a electron in a form of proton. H positive is a proton. Either you are losing it or you are gaining it. Okay, this is a redox reaction. In a very simple definition, either losing or gaining a proton. Okay, so this is a proton, H positive. So whenever you see the name dehydrogenase, you will see NADP, you will see NADPH. When you are gaining, you have this. When you are losing, you have this. Simple concept. Don't mix anything. Very simple concept. So now they have given you the example of pyruvate dehydrogenase, which is a enzyme that converts pyruvate into acetyl-CoA. Okay, this is not a rate limiting enzyme. This is just an enzyme that is converting pyruvate, the final product of glycolysis, into the first substrate of TCA cycle, acetyl-CoA. Okay, let's see this in your Lippincott page number 5. कैसे आ गए? So we will study till TCA cycle. Our last will be TCA, and We have done like 40 minutes, 2 classes. You have to study by your own self also. Please revise it, okay, and learn it every day. Where is page number 92? Okay, here. So this is my favorite page of this book, page number 92, Lippincott. Okay, this is the pyruvate, the last step of TCA cycle, and this is acetyl-CoA. In this step, you use this enzyme pyruvate dehydrogenase. Okay, so this is the step, but here it is not written as big, and they have not shown the energy molecules over here. But I'm sure in your first aid book, like they have in your first aid, they might have shown this. So you can also see in here, pyruvate is converting to acetyl-CoA, and see this number 14. This number 14 is pyruvate dehydrogenase. Okay, so pyruvate is converting to acetyl-CoA and the enzyme is pyruvate dehydrogenase. This enzyme is dealing with the reaction which we called redox. Now I just want to show you that what is going on in a redox reaction. See this pyruvate dehydrogenase complex. We will be studying it in detail, but you only see this reaction right now. See this reaction, so it will help you understand. Let me make it over here for you. With this is pyruvate and this is acetyl-CoA. Okay, and what is happening now is you are converting the pyruvate into acetyl-CoA. So what are you adding? You are giving in NAD positive. Okay, that NAD is converting into NADH. I told you a proton. Okay, wait a second. Okay, NAD is converting into NADH. Okay, you are also giving a coenzyme A, okay, which is added to acetyl group, and in return carbon dioxide is released. So this is a step. This is a whole step. Okay, this is the reaction going on. So the enzyme is pyruvate dehydrogenase. We write it PDH. So this PDH is always dealing with redox reaction. You will always see this NAD positive and NADH over here. Okay, so if I take you to the TCA cycle rate limiting step from this book, okay, so wait a second. If I take you to the rate limiting step of TCA cycle, glycolysis, TCA cycle, isocitrate dehydrogenase. See this picture. Just see this picture. What's going on is, you see acetyl-CoA. This is your acetyl-CoA, which was converted from pyruvate. Okay, it gets attached with your oxaloacetate because this is a cycle, right? So if this is acetyl-CoA, here it is oxaloacetate. So oxaloacetate comes and join with this. Okay, and then they convert to citrate and then isocitrate and then alpha ketoglutarate. So this step, which is isocitrate is being converting to alpha ketoglutarate, this step is the rate limiting step, because this enzyme isocitrate dehydrogenase is a rate limiting enzyme. Okay, and it is a dehydrogenase. So you see NAD is converting to NADH. So everywhere you see dehydrogenase, you will see this redox going on. This is called redox. You are, one substrate is losing the electron, the other one is gaining. So this losing and gaining of an electron in the form of proton, which is H positive, is called redox reaction, and dehydrogenases are always dealing with redox reaction. So everywhere you see dehydrogenase, you will see this work going on. And now what you see over here is, because I told you TCA cycle generates ATP, okay, TCA cycle makes more ATP. So if there is more ATP or more NADH, this cycle will inhibit. But if there is less ATP, or you can say low energy molecule like ADP or AMP, okay, this cycle will promote. Also, calcium has a role in promoting this cycle. Okay, this is important. Please remember, calcium plays a role in promoting this cycle, because TCA cycle gives you more um, energy, and this ATP is also utilized into your muscular activity. Okay, so calcium will promote it more. So kindly remember this calcium, because it is a different thing. Okay, and they can ask you. So if I take you back to your textbook of first aid, you see this step, isocitrate is converting to alpha ketoglutarate. This step is a rate limiting step, and this number 18 is isocitrate dehydrogenase. Okay, so here you see TCA cycle, isocitrate dehydrogenase, and you have more ATP or more NADH, you will inhibit this cycle. You don't want this cycle, because you already have high energy molecules. But if you have a low energy molecule, this cycle will promote. So it is the same concept as glycolysis, as glycogenolysis. Same concept is applying over here. But the difference lies in, you see NADH the very first time. You haven't read NADH anywhere. Abhi tak you haven't read NADH. Okay, so please remember, dehydrogenases deals with NAD and NADH because they are redox components. Okay, and dehydrogenases deals with redox reaction. Please remember this. Okay, and please remember the name of enzyme, isocitrate dehydrogenase. It is converting citrate into alpha ketoglutarate, isocitrate into alpha ketoglutarate. Okay, please remember this step and this enzyme, rate limiting enzyme. Okay, now we are done with these 5. Okay, and we have done with few enzymes. So we will continue this lecture tomorrow. We have done these 4 enzymes and we are left with the rest of them. So I will explain you the functions along with the cycles, okay, so that it will be easier for you to remember and understand. So whatever you have learned till now, including the ethanol metabolism of yesterday, please revise these pages. Okay, you have to you have to keep revising your book again and again when you have time. Okay, otherwise you'll forget it. So we will be dealing with our next lecture, okay, in some time, and I'll post that in the link in the group. And this is all about today's lecture. Okay, if you have any questions, you can ask me. We are left with like 50 seconds and the recording will stop, or the lecture will stop. Okay.